Our next speaker is from Uruguay, Ana Umpiérrez. And Ana is a PhD student at the Clement Estable Institute for Biological Research in Montevideo, Uruguay. She has interest in microbiology, and she will speak to us on genetic diversity and antibiotic resistance of E. coli isolates in neonatal calf diarrhea cases. Let's welcome Anne.
Okay, thank you. As he says, I'm going to present the work Genetic Diversity and Antibiotic Resistance of Escherichia coli isolated from neonatal calf diarrhea cases in Uruguay. This work was performed at the Department of Microbiology at Instituto de Investigaciones Biológicas Clement Estable in Montevideo, Uruguay, at the Department of Bacteriology and Virology at Faculty of Medicine in Montevideo, Uruguay, and at the Laboratory of Immunochemistry and Biotechnology at the Faculty of Veterinary in Tandil, Argentina. Just to illustrate where I came from, Uruguay is a small country in South America. It's about 3.3 million people, and our main economic resources are agriculture and livestock. And in terms of numbers, bovine stocks is about 12 million heads from dairy and beef production, and in terms of export, beef export reaches $2,000 million last year. As all you may know, neonatal calf diarrhea is one of the most important infection diseases of dairy and beef calves around the world. It is associated to high economic losses, and it can affect young cattle between 12 hours after birth and 35 days old. This disease is very common when intensive systems of production are applied. These systems increase the efficiency of the production, but on the other hand, decrease the health status sometimes due to, for example, stress of the animals. Etiology of neonatal calf diarrhea is very complex. There are several pathogens associated to it, for example, rotavirus, coronavirus, Escherichia coli, Cryptosporidium parvum, and Clostridium perfringens. And E. coli is going to be the focus of our study. Okay, for the prevention and treatment of this disease is essential, for example, the correct intake of colostrum of the calves, the good management practices, the nutrition of the animals, the hygiene of the establishment, sometimes the use of vaccines, and in some cases, the use of antibiotics. Antibiotic therapy has been used for long decades, and as you can to treat different infection diseases in animal and human, and as you can see, the families of antibiotic uses in animal and in human are the same. However, misuse of antibiotics has increased the occurrence of multidrug-resistant strains in the last years. And this is more important when we take into account the occurrence of plasmid-mediated quinolone-resistant proteins and transferable extended-spectrum beta-lactamase strains. Neonatal calf diarrhea is present in Uruguay, and previous work in our laboratory has established that there is a high prevalence of E. coli adhesion-related genes like CLPG and F17, and low prevalence of F5 genes and toxin genes. However, there is no records associated to E. coli antibiotic susceptibility, detection of resistant genes, or genetic diversity studies of this pathogen in neonatal calf diarrhea. So the objective of our work was to evaluate the genetic diversity, the clonal distribution, and antibiotic susceptibility patterns of a collection of E. coli isolates recovered from calves in Uruguay. First of all, to study the clonal diversity, we performed ERIC PCR. We employ a E. coli collection of 303 isolates that was previously characterized in our laboratory. And then we select 22 isolates to perform multilocus sequence typing. Also, we studied the antimicrobial resistance. To do this, first we studied the reduced sensibility to ciprofloxacin, at the disk diffusion method, and molecular detection of transferable genes for plasmid-mediated quinolone resistance and extended spectrum beta-lactamasas and the presence of class 1 integron. Well, after ERIC PCR results, we could observe that there is a high genetic heterogeneity among the whole collection. As you can see, in green are isolates from healthy animals, and red are isolates from ill animals. This is a tree representative of one department, like a state, called Canelones, but we observe this pattern in the whole country. And we could observe that NCD and healthy isolates grouped together. Then, after MLST analysis, we could detect 17 different sequence types in Uruguay. 4 of them were new sequence type, and 13 were previously described. Only one sequence type 101, was found in 2 isolates from a single farm, one from a healthy animal and one from an affected animal with neonatal calf diarrhea. If we look inside of this previously reported sequence type, we observe that only 4 were exclusively associated to sample of animal origin from septicemia and mastitis and were of a healthy animal, and 9 of them were associated to sample of animal and human origin. Particularly, we would like to highlight the occurrence of sequence type 21, member of sequence type 29 complex, which is associated to high virulent E. coli isolates, the presence of sequence type 69, which is associated to human uropathogenic Escherichia coli, and 4 sequence type associated to extended spectrum beta-lactamasas. Again, when we studied the diversity of these genes in MLST, we observed that there is a high genetic heterogeneity among them. Then, when we studied the reduced sensibility to ciprofloxacin, we observed that 40% of the whole collection were resistant to this antibiotic at that concentration. So we select 112 isolates to perform this diffusion. And we observed that 83 of them were resistant to ciprofloxacin disc. Moreover, we detect that 71% of these isolates were multidrug resistant to antibiotics. In terms of co-resistance, for example, we highlight that almost 10% of the 100% of the isolates were enrofloxacin and ciprofloxacin resistance. Almost 80% were resistant to ampicillin and ciprofloxacin, and almost 70% were resistant to cotrimoxazole and ciprofloxacin together. Molecular analysis allows us to detect transferable genes. 7 isolates present plasmid-mediated quinolone resistant genes, and 3 isolates were extended spectrum beta-lactamasas. And finally, 5 isolates of the 22 studies in this case were class 1 integron positive with this genes arrangement, which allows them the resistance to aminoglycosis and trimethoprim. So this work was the first molecular characterization of E. coli antibiotic resistance genes of bovine origin in Uruguay. We detect high numbers of isolates resistant to fluoroquinolones, including enrofloxacin, the occurrence of multidrug resistance isolates, and the presence of extended spectrum beta-lactamasas isolates. Also, we detect the presence of plasmid-mediated quinolone resistant genes. This work was the first detection of QNRS1 in Uruguay. Also, the diversity analysis showed that there is a high genetic heterogeneity among E. coli collection, with same genetic variants in healthy and in animals. And MLST analysis allowed us to identify 17 different E. coli sequence types. Particularly, we would like to highlight, again, the sequence type 69 and sequence type 29 complexes, worldwide distributed in bovine and human, and highlight the zoonotic role of these isolates. So, as a conclusion, antibiotic resistance is a relevant problem in livestock in Uruguay, and these results should be taken into account when new prevention and management strategies are planned. Also, we determined that genetic heterogeneity highlights a complexity scenario of neonatal calf diarrhea in Uruguay. we observed that there are potentially pathogenic E. coli isolates that can cause disease in humans, and the resistant profile could play an important role in zoonotic diseases caused by these strains, favoring multidrug-resistant strain spread into humans. This work was supported by PEDESIVA, the postgraduate program in Uruguay, and by the Agency of Innovation and Investigation, ANI, who found my PhD grant and a project for young researchers. Thank you for your attention, and visit Uruguay.
Do we have any questions for Anna? So there's one on the side there.
Scott McDougall from New Zealand. Have you got any data on antimicrobial usage on these farms in terms of, I mean, you're demonstrating ciprofloxacin resistance there. Do you know if fluoroquinolones are being routinely used for these calf diarrhea cases?
I can say that fluoroquinolones, and particularly rofloxacin, is widely used. I don't know if in the particular farm where I took these isolates, but are frequently used.
Anna, you identified at least one strain or one type that has zoonotic potential there. Has that been a problem in Uruguay, say among workers on farms?
Well, this was the first work that identified that. I can say that it's a problem, but this is a start to study if this is or not a problem. Okay,
I was just wondering if any of those strains have been identified from humans who have worked on farms. No.
No, there's no record about that.
No. Okay. Any other questions? Okay, well, thank you very much, Anna.